Hi everyone, my name is Natalia Derogi and I am a freshman in the School of Engineering here at Columbia in a part of CORE. This next panel that we have up is called Spinning Out in Control, Life at the Center of University Research and Startups.
Great, thank you. Can people hear me okay? Great. So I'm not in the program, I'm subbing in for the moderator, so I'll just introduce myself very briefly and then I want to get to our esteemed panelists today. My name's Oren Herskowitz. I run Columbia Technology Ventures, which is Columbia's tech transfer office. So when the faculty researchers, or the graduate student researchers, come up with a new scientific invention, they tell our office about it, which happens about 400 times a year across the university. Our office works with them to figure out whether the idea has commercial potential, and if so, we file patents where appropriate. That happens about 250 times a year. And then we work with them to either find a big company who wants to license that idea and turn it into a product or service, or increasingly, so that happens about 100, 110 times a year. And then increasingly, many of our faculty and graduate students want to start companies of their own based on their science, as represented by the panel we have here today. So typically, that's around 20 to 30 startup companies a year based on research emerging from the university's research labs. So I do a bunch of other stuff. I teach a class called Intellectual Property for Startups, and I do a variety of work on the city, state, and federal level on entrepreneurship. But that's enough about me. I'd love to ask each of our panelists today to introduce themselves and just how they got to Columbia, a little bit about their work, but in a way that we'll all understand, hopefully, myself included. And then we'll move on to the topic of startups and entrepreneurship. So why don't we start with McCall Lipson.
So I've been here 2.5 years. I came from Cornell. I started there a group that works on silicon photonics. This is a field that we pioneered, basically taking light and optics as you know it and miniaturizing on a tiny little chip, just like we do with microelectronics. So today, optics, like imagine if you have tons of fibers and lenses and everything, all of these could be miniaturized and massively fabricated on a tiny chip. So this has tons of applications, and we've patented a total of 40 patents. And several are now being licensed, and we have now a new startup as part of Columbia to do LiDAR, which is actually very related to the previous talk that we've seen, sending beams of light from a tiny little chip, getting them back, and from that reflection, telling where exactly the car is located. So for autonomous vehicles. So we're very excited.
Right, that's great. We'll come back to that later because I think your work has such a broad array of possible uses that it's sort of interesting to think about that as an entrepreneur. But great, thank you, McCall. So Tilla Wargal.
Hi, my name is Tilla Wargal. I've been at Columbia for 22 years. Started out as a postdoc in a completely different field that I'm having my startup in now. And I'm originally an MD. I still practice as a clinical pathologist. And the startup refers to actually a project that started just out of pure interest. We were postdocs. There was a whole group of postdocs, and everybody comes from a different lab. And over the years, you sort of run into each other again, and you know that people do stuff that is interesting. And me and my co-founder, we were friends, we remained friends, and we started talking about many other things, and then understood that his technology, DNA-based aptamer recognition sequences, really have something relevant for clinical pathology. And in the beginning, it took me a while to understand that with just a small piece of DNA that you can manufacture very cheaply, you can accomplish a lot. And his specific interest was small molecules. Is also at the first sight not such a great theme. You always want to talk about something big, but small molecules is a real challenge in diagnostics. And so I learned the technology. I was fascinated by it because the real key moment was like 15 years ago. I was exposed to that technology. I understood the technology. I worked on it with my own hands to see, does it really work, or is that just more basic science? Does it really work for a human matrix? And that's basically when we started. Sort of didn't have much response because the molecules we were working on were so uninteresting for other people probably. But we then switched to other themes that were really addressing unmet clinical needs. And at that point, you really have almost like an obligation to follow through with something. So we focused on small molecules, amino acids. That is not as relevant to the majority of us, but it's very relevant to people with inborn errors of metabolism. And basically what we managed to do is to supplement. We have a technology that you don't need a mass spectrometer for. So instead of using a mass spectrometer at a central lab, you can now do the analysis at home in a very cheap way. And that's basically what UpTheTech does. And you can expand it to other molecules, et cetera. So that's what we do.
Thank you, Tilla. And Angela Cristiano, I've worked with Angela the entire time, the whole 12 years I've been here at Columbia, one of our more prolific inventors and serial entrepreneurs. So maybe a bit about yourself and maybe just mention the companies that you've worked on.
So I've also been here 22 years. I didn't know that. This is still my first job. I came here after my postdoc, and I never left. So I have a PhD in genetics, and my appointments here are in dermatology and genetics and development departments. My lab uses genetic techniques to understand the basis of skin and mostly hair disorders, particularly hair loss disorders. And then we try to figure out ways to use the genes that we find as potential therapeutic targets, both for hair removal and as well as for different forms of hair growth. And so over the years that we've been doing this, I've had the good fortune to establish 3 startups now. My first one was called Skinetics. It was founded in the early 2000s, around the time that siRNA was exciting and discovered, and Skinetics used an siRNA approach to target genes in the hair follicle with the goal of treating for hair removal. Just about as soon as it was formed, it was acquired, so Skinetics was fortunate to get an STTR grant, and then was acquired by an shRNA company called Sirna, and a few years later Sirna was acquired by Merck, and so that was my first one. So I didn't actually get to have the startup experience because it left quickly.
Although a high-class problem to have.
High-class That one sort of, yeah. The second one was called Vixen, and that was just a few years ago, and Vixen was focused around small molecule targets for alopecia areata, which is an autoimmune form of hair loss. And very similarly, as soon as Vixen went out sort of trying to get funding, Vixen was also acquired by another company called Aclaris, which is one of the larger dermatology development companies. So that one, too, I didn't quite get to have the startup experience. But the third one is called Rapunzel, and that's the one that we're working on now. The focus of Rapunzel is to use autologous stem cells, your own hair follicle cells, to grow new hairs. And so Rapunzel just got an SBIR grant, and hopefully this one will actually get to have the startup experience and actually spin one out. So those are my 3 stories, and I'm sure we'll talk more as the panel goes on.
It's interesting to think about where the idea to start a company around this stuff comes from. And I think Angela's work in particular is probably the technology area where we get the most inbound inquiries from people saying, when can I get that hair replacement drug? Or when can I get the hair removal drug? And so I'm actually curious, as each of you were thinking about the path you took, how did you know you had something that was, of all the work you're doing, of all the, especially I know in your work, there's dozens of different areas that you and your students are pursuing. How do you know which one to start with? How did you know when you had an idea that was going to make for a great startup? And maybe I'll start with you, McCall, but if each of you
So in my case, I think I'm fortunate because there is much less subjectivity. I mean, it's in all these areas where my technology is applied, like sensing and data communications, computing, LiDAR for autonomous vehicles, even all kind of light emitters, for example. Just very, very efficient bulbs. There are always very clear numbers that you need to hit, and these are known. So there is no subjectivity. If you hit those numbers, you're good.
And when you say numbers, maybe explain.
So when I mean numbers, energy efficiency, or if it's relevant, speed, or in the case of LiDAR, how far can I go? With what precision? So all these numbers, these are tables, and we know when we got something really good.
Interesting. And I'll just ask a follow-on question before we move on. One of the things, how many folks in the room have gone through some kind of a lean launch-based, like a lean launchpad-based accelerator or iCore or something like that? Or familiar with how those work? Customer discovery? So the whole concept of customer discovery is to go out and talk to your customer and figure out, do 100 customer interviews, figure out what people really want. Does the fact that your startups and your science tend to rely on such clear objective measures, is it still important to go talk to the customer, or is that secondary, do you think?
I think it's important because when we get out, we don't yet have the numbers. We have we have projections for these numbers, but we need to know how close do we need to hit them. Are we in a good place relative to what the customer needs or not? But we know very clear where do we need to go.
Okay, got it. Tilla, how about you? 22 years, and this is the first time you've decided to go jump in. Why now?
Well, I would have jumped in 10 years ago if somebody had believed me. But so here I am. How do I get to this? I agree with Michal. You have numbers, they work, it's correct. Whoa, it's much faster, much cheaper. That's basically what got me started. So I used to be a director of biochemical genetics, where basically you handle clinical samples for people who have diseases. You use mass spectrometry, you do regular routine stuff, but it takes forever, and it's very expensive eventually. And you know you can do this in 30 minutes with the other technique that you transfer over from the more basic science end of Columbia. And that's very tempting. You can almost not do it. Not do it. I mean, yeah. So, it's a progress that needs to get out there. Yeah.
Great.
So, in dermatology, the category of disease that has what we call the most unmet medical need, meaning scarcity of treatments, is actually hair loss. And so there was no question in our field how important new treatments for hair loss are. But to take a step back from that, the reason my lab works on hair loss is because I came to Columbia and 6 months later my hair fell out. I developed alopecia areata myself, which is an autoimmune form of hair loss, and looking around and seeing how little was known at the time was really what launched our lab down this path of hair loss research. But what was the surprise for me was the outpouring of, as Oren mentioned, emails and letters and phone calls from patients asking, in some cases demanding, these treatments be pushed forward. The surprise was the outcry of the need for hair removal, as well as for hair loss. I think for hair loss, it's a given that most people want more hair, but I was shocked at the outpouring from people who actually suffer greatly from having excessive hair. And so Skinetics really got started basically as a push from the community of patients who really suffered, and that was unknown to me. So ours came kind of two ways, from my own experience, but then also from listening to these the communications from people, from patients, your customers, or people who wanted it. Yeah.
And when you were thinking about the question we at Tech Transfer Office often get is, how do we decide whether you should license something to a company or start a company around it? And the answer we typically give is, it really depends significantly on what the faculty want to do and the graduate students. But it's really very technology specific. So I'm curious, when you think about the inventions coming out of your labs, which are quite numerous for all three of you, how do you think about whether each one of them should be a startup, or whether you'd rather we just go license it to somebody? Like, is that even something that crosses your mind?
For me, I must say I'm not good at the business end, so I basically listen to what CTV tells me.
Can we get that on camera?
I hope you're better than me at the business end.
I guess so for me, it's almost technology specific, and it's also a balance of how far we can take something in the lab versus how soon we need to collaborate. And in most of our cases, we need to collaborate relatively soon. We need medicinal chemists, or we need a GMP facility for the stem cell work, or we need access to expensive, reagents or nucleic acids in one case. So for us, it's sort of a titration of how much we want to stay as academics versus if we ever want to take the plunge and cross the street over to the Audubon building to actually try it. And so in our case, it's always been an early decision to partner in one way or another. I think both of ours were accidental. We would have definitely preferred, I think, to try it, but partners came along that were, interested and very different. So in the first case, we had very little collaborative involvement, and suffice it to say that once you've done it badly the first time, you learn, so the second time you get it right. So the second time with Vixen, we made sure that we have, a close relationship in terms of consulting, advising. We're really there every step of the way. And in terms of the collaborations, with Skinetics, it was founded with a colleague, one colleague inside, two outside. With Vixen, I had a co-inventor, Raphael Klein's former faculty member, co-inventor, and so rarely do we do things, alone. So you also have to have consensus among your team of PIs. What's the right course? So.
How many graduate students in the room? Graduate students or postdocs? Okay, so this question's for you. So as these inventions are emerging and they make the transition out into the world, either through a license or a startup, what role do you as faculty members typically have in that? You hear about the faculty who will leave the university and go take a management role. In our experience, much more often the faculty member stays at the university. And students tend to play a very important role in that bridge. Can you talk a little bit about what your experience has been, or you think will be?
Yeah, this is definitely my case, specifically with the LiDAR technology that we are developing. So basically, the students have ownership. They are doing it. They are pushing it, and then often the VC or the partner that is interested in the technology, they want to talk to me at some point. And we try to push it, and we have also quite a lot of internal discussion between us, what path should we take. But the students are taking ownership, and it's amazing. They should be like that.
The name of the startup, by the way, you should definitely watch this one. It's called Voyant, and the two students who are behind it are just absolutely phenomenal. Yeah. We had them
Chris
pitch.
Fair and Stephen Miller.
They're amazing. And actually on the brink of some rather significant fundraising.
Oh, absolutely. Yeah,
so,
it's going to be very big.
Yeah, we'll come back to that one in a moment. How about you guys? How did you think about what role you wanted to have in the startups you're involved in?
So, if you work with others that fund it, they have, of course, their vision. And I personally liked, in our case, that it was already on such solid feet that I was able to let it go and knew if it was done outside of the lab, it works, because that's something that was very important, obviously. I don't really have an opinion about this. I mean, I'm going to launch a second one where I hope to have exactly the role for a student who needs to graduate first, and he will. And so ask me next year. It's very difficult to. If you come from academic research, you work together very closely, and all of a sudden you enter into a world that is business-driven, with people who don't have that same attachment to the research you do. So for me, that is always not as comforting, because I'd like to see it through, like to make it the way I envisioned it. But that's the name of the game. You know?
I think, again, for me, all three experiences have been different. The first time we were dealing with partners who were not dermatologists, didn't understand skin or hair diseases, and we always felt like we were sort of having to justify our existence. Why is this important? And it was really starting from a very limited place. So I think you have to sort of get a feeling for your relative value in the configuration of people that are already there. I think there it was made clear to me early on I wasn't too valuable for the collaboration. But the second one was totally different because our partners are dermatologists. We didn't have to spend any of our time justifying our existence. They understood how important hair is, how important alopecia is within the realm of dermatology. Now it's sort of in this configuration. I'm a valued advisor because I understand the disease well, both as a patient and a professional. So my opinions are more welcome and often called upon. In the third case, though, in Rapunzel, so both of the first two of mine were so far outbound that they really was like a runaway train in a way. We didn't have time to populate either with much of my time, let alone anyone in the lab. Rapunzel, I hope, will be different because it's really incubated a much longer time in the lab, maybe 10, 12 years of work. We do have some interested folks now in the group that may be wanting to step out into it. So I hope this one maybe takes more of that configuration. And in some ways, this is the one that's I would say, the most reliant on expertise in the lab. So I think it's kind of a balance of the PI's value to the team and then the technology and how dependent it is on the knowledge, the collective knowledge that's in the group versus in the company or in the partner.
So actually, let's talk about that. Each of you and your students bring technical expertise and obviously the initial innovative idea. But as you were thinking about launching the company, or even seeing whether the idea was a launchable product, how's it been being at Columbia? What's the ecosystem like here from your perspective? What resources did you rely upon that you found particularly helpful? Any advice to give the folks in the room about things they should do when they're thinking about launching their companies up here on 116th or 168th Street? Sort of like, what's it like here?
Maybe I can talk. I actually have a good perspective, right, in the sense that I'm relatively new to Columbia, and it's been amazing in the sense that, I'm always overwhelmed, and in terms of time, and you guys take over in terms of you tell us exactly what we need to do, and kind of guide us. And you come back. You don't let me run without coming back and following up. And one very good, like a striking experience that I had that I really never experienced as strongly was every patent that we submit, every provisional patent that we submit, there is a follow-up where there is kind of a pretty deep technical discussion on where could this be applied, what is a competing technology, and it's a wonderful.
Great. And you work with Greg Maskell in our office.
Yes, and Greg has been just wonderful.
Great.
So I have a longer history at Columbia, and my first experience was, so you write a patent, you're all proud, that's great, you believe in this, and then nobody wants it, and so that's the end of your patent. And then you come up with a second idea, oh, you again, yeah, it's brilliant, and this and that. But eventually, you hit it. I still think these first ones were brilliant, except nobody else understood that. Doesn't matter. You then have this patent, and what really helps is, what really helped me, we have these incubator sessions here, Coulter grants, it used to be called. I received a prize there from Coulter twice.
It's now called the BioMedX, or Biomedical Engineering Accelerator. It's run out of the engineering school in collaboration with the medical school and others in our office too. But
and that is really helpful. So anybody young in the audience, go there because you see a lot of people who are on the you come from basic science, you rely on data, and everybody knows how to interpret a curve or some kind of other formula. But if you need to bring your idea across, you need to communicate with the investors, and you learn that there. And you also get comfort in how clumsy some people are, that there's hope for you. You will start out clumsy and you come out somehow good. And I religiously followed this, and I was actually amazed. I was not the only one who followed it. Young people and the more advanced, more established people, they all came and listened. And that really helped because it puts it in perspective. And, my technology apparently was good enough to be noticed. But even those that didn't have noticeable technology really learned a lot from there. And you also form relationships. You understand, or you want to be at the point that people who work at CTV understand that your data has substance and that you're reliable and that you're potentially a good partner, etc. So that is very, very helpful.
Great, thanks.
So one hard lesson to always learn, I think, when you're just starting out is the importance of filing and filing early. So anytime, you think you're going to a Gordon conference, so you're just going to put an abstract, or you're going to this meeting, so you're just going to say something here on a poster. Every time you do that, you're basically ruining your chances of patenting. So one of the most important lessons is learning to do that early enough so that you protect any public disclosures, but also not too early that you start burning your 1-year clock, right? So as Tilla mentioned, you have 1 year from the time of filing your provisional until you come back a year later, and the university works with you to decide if you're going to convert that into a PCT. That's the point where you need all your data, right? So you need that year to kind of, really firm up your data, hopefully publish your paper, but you can't do it too early, and you certainly can't do it too late. So learning when is the right time to pull that trigger is important. Then you have to back up and do the math that Oren mentioned at the beginning. I'm not sure if everybody caught it, but he said they get about 400 invention reports a year and only file on about half of those 250. Yeah. So that means there's another internal selection process that goes on inside the university, and that's not always on paper, right? So oftentimes you get called in and you're asked to present the work either on PowerPoint in front of the technology transfer group or the filing firms, the law firms that we use to file. So that's your first pitch right there. So you're competing against other Columbia colleagues to get your work filed on, and that's where you really learn to do it yourself. So before you even put that invention report, right, you write your invention report as if it's almost your pitch deck. You've already gone to Google Patents and you've looked up what else exists. What's the prior art? You learn to do the prior art search yourself. What's in the literature? How am I going to thread the needle with this invention to avoid what's already out there, but to create novelty? So it's your job as the inventor to kind of help lead the discussion in the CTV office together with the officers there to find a way, if there's valid invention there, to protect it and to bring it forward. So for me, that was one of the hardest lessons of, like, you're already ABC, always be closing from the time you set foot in the CTV office. That's your first pitch. So it's really that early that you kind of have to, balance the timing of that. So you need another 3 months really there, at least, for that process to take place. So you've got to kind of be very conscious of the timeline of both the CTV process, the filing, and then when your data is needed, if you have to graduate or if you have a grant coming up or whatever. So I think that those early steps are very critical, obviously. If there's no patent, there's usually no startup and no license. So it's the most important part.
Maybe I should add one more point to Angela's experience. A mistake to avoid from personal experience is when you actually file your patent, really think big. Help when you are discussing with CTV, think big in terms of where could this be applied, and even more especially if you are doing anything related to nanotechnology or physics. Where could this? What kind of configuration this could be in? I have several patents that are just so easily circumvents that they were never licensed, and they are the basics for whatever people are using today, but they're very, very easily circumvented.
I should point out that those were done at a different university.
Yes, they were.
Prior university.
No, but it was my mistake. It was my mistake, though.
And that's great. And actually, that wasn't meant to be an entirely self-serving question about our office, although I'm happy it was. But beyond that, like Angela, I know you've worked with students at the business school previously to help with your research, and to fill out what McCall was talking. I'm sorry, actually, what Tilla was talking about. Columbia runs either on our own or with our peers at NYU and Cornell and others a variety of what we call lab to market accelerators, which are programs to help faculty members and their graduate students take their nascent ideas, these incredibly exciting inventions, and learn how to start companies around them. So there's 3 of them around life sciences, BioMedX, the Accelerating Cancer Therapeutics, and the Translational Therapeutics Resource Accelerator. I'm possibly getting those wrong, but you can look at our website. And then there's one in clean energy called PowerBridge New York. So if any of you are energy-related entrepreneurs, Columbia can be helpful there as well. And the last one is, for now, is around media. So if you do new media, augmented reality, virtual reality, there's also help for you at Columbia.
There's also, so I just wanted to give a perspective. Over 20 years, the ecosystem here has just grown exponentially. There's so much more available now in terms of resources and programs and funding. But back when I started, there was also a class in the business school called Launching New Ventures. I don't know if it's still given.
It is. In fact, David Lerner, who's here, teaches it, I believe.
Hi, David. Yeah. I know David. So that class is great because they'll pull from the medical center as well, because the students in that class need a technology to focus on their semester. So you get a group of, business school students to work on your project for the whole semester, and that used to roll into the greenhouse program. I don't know if it still does, but so there's lots of opportunities now that didn't exist before, and they're all super, super helpful. Then after you get the patent part, then you have to learn how to write your executive summary, your pitch deck, and then your business plan, which is like writing an R01. So any help you can get with those documents, being, again, scientists, a lot of that doesn't come naturally.
Right, and if you guys have any questions, by all means, go to entrepreneurship.columbia.edu. It's a great resource for all of the resources available for you at Columbia and across New York City. So definitely recommend you check it out. Let's talk about money for a little bit. So each of you have been successful or are about to be successful in fundraising. Tell me a little bit about how you found your investor or investors. How did those relationships come about? How challenging was the process of raising the money? Did you feel like it went smoothly or is going smoothly? Any lessons learned from the audience? Like, how do you go out? These are very hard to do without money.
Well, CTV had a major role in that. We got introduced to investors. We, in the beginning, now kind of the word is out, and we are being contacted by multiple, which is, you always know that it is important to have a perspective and know exactly your market value, but it's, we definitely are seeing this now.
There's this is basic economics, but the math changes substantially in terms of the value if you're negotiating with one party versus negotiating with 2 people. So nothing brings clarity like competition. It's a great situation. You guys have had your dance card is very full right now, so it's very exciting. How about you?
So I participated in we participated in the Coulter, and there was a company, an investors group that is interested in being with young, hopeful companies and developing them further along. The monies that go in there are high, and for people who have previously only worked in science and on grant-related budgets, it's very out of my league, because a lot of money goes also to lawyers, to stuff you never imagine, and you're really not an expert in. And CTV again comes in very handy as a guidance for this. In the end, you have to go out and raise money yourself, or for that company, of course. And I must say, if you have a good product and you have the data to show, that is not so difficult. But to get somebody else's money is always difficult enough. Yeah.
So we call this person, this mythical person, the unicorn. So the unicorn for us has been the person that kind of joins the team early and is the catalyst person between what we do and helping us either get introductions to that money or facilitating in some way. So our first deal, Kinetics found its unicorn because we did such a bad job at one of our VC pitches that we were out in the hallway sort of crying, and it was a disaster. And one of the partners that we presented to came out and said, "You guys were horrible, but I love the idea, so I'm going to help you anyway." Which was like
okay, so the lesson is be really bad in your first pitch, and then.
And it changed everything. He did, and he was our unicorn. It's kind of like Shark Tank, only worse. The second time, though, we were very helped by our field, and I don't know if this pertains to you guys as well, but within dermatology, there's a catalyst called Advancing Innovation in Dermatology, and it's a group of sort of derm entrepreneurs that organize themselves into this wonderful catalyst. And that's exactly what they do, is they help people who have early stage technologies be introduced to either the community or company partners or people that can give them drugs or chemists or whatever, free of charge. They're just amazing ex-pharma folks, in some cases current pharma folks. So they helped a lot with Vixen, and now for Rapunzel, it's a whole different question now because our needs are different. We're talking about stem cells and GMP, and this is a brand of unicorn that we haven't really tapped into yet. And so in our experience, one of the key ingredients in the early stages has been a business savvy, friendly person that can kind of come in and help open the doors and understands the science and values the science enough and is themselves not greedy and willing to work basically on spec as part of creating something of greater value.
For those entrepreneurs in the audience, there are a number of resources around Columbia where you can tap into that entrepreneur network. I know the business school has a great entrepreneur in residence program. Our office at CTV has executives in residence, 5 or 6 per semester, who are under non-disclosure agreements, so you can talk to them freely, and they are free. They don't take any equity or cash. And they're there to bounce ideas off of. All of them are serial entrepreneurs or funders or industry veterans. And we're actually launching a program shortly across all the academic medical centers of New York City called the New York Life Science Venture Exchange, which the goal of which is to connect those unicorns with high promise startups based in New York in the life sciences. We've got a similar model going for clean energy that's getting up to a good start. So there's a lot of, please. No, it's okay.
I guess the other thing that's changed too in the past 10 years is that there's so many more pots of local money. So there's $100,000 grants basically every time you turn around. In our case, we went to STTR and SBIR, but you don't have to go that far. There's so many, Oren, you know them better, but local, development funds now that didn't exist before that are easy to at least find out about and then to compete for.
Sure.
Yeah, I agree with this. At the medical center, there's a preventive precision medicine initiatives, and so just participating is great networking. If you get a grant, it's not a lot of money, but it is very good to give you recognition and to connect you to people who value that you've achieved a grant, on that topic. So that's very helpful. Right.
Yeah. How many folks in the room, when Angela mentioned SBIRs and STTRs, just show of hands, how many people know what those mean? Okay, less than a third. So can we just do 2 minutes on SBIRs? Because it's free money from the government. It's please, go ahead.
So I guess, I don't know how long ago, 20 years ago now, the NIH was mandated by Congress to put aside 1% of its annual budget for business development to translate many of the technologies that were discovered under NIH grants and tried to reinvest that into the community. So SBIR stands for Small Business Innovation Research, right? Yep. STTR is, Similar but different. Right? The main difference is in STTR, the grant holder can be at the university. The SBIR, the grant holder has to be in the business side. So there can be subcontracts in either direction. The STTRs are much harder to get. There's a smaller pot of money, and they're much more competitive because you're competing against your own colleagues. The SBIRs are actually much easier to get because there's a lot of people, at least in the hair loss world, trying to do really bad research. So that helps us. We look better against the backdrop of not so great science. So these programs are out there. They're 1 year for $100,000, and then you can convert to second year and third year for $750,000. And then if you're really lucky in your area, sometimes they launch over $1 million. So you do need a few things to get started, though. You have to be formed, so the business has to exist. It usually needs to have a space, and this is where the Audubon Building, Harlem Biospace, a lot of the other incubators come into play. But the good thing is you can use the grant to pay your rent. So the grant is pretty liberal in terms of what it can cover for the business: legal costs, accounting, rent, overhead, et cetera. So these are programs, again, through the NIH. They're a little bit more cumbersome, but I will say very prestigious. So when you're out talking to investors and you have an SBIR grant, it's a mark of excellence.
Yeah.
So they're worth the trouble.
And actually other agencies, the NIH has it, but actually every federal granting agency is required to put aside a certain percentage of their budget for these. So the NSF has them, DARPA, ARPA-E. In fact, we have one of Columbia's startups, Lumiode, which is an NSF-funded startup that got an SBIR grant from them as well, is now just raised their big A round, and they are based in the Mink building just down the hill in Harlem. So actually, maybe let's touch on that for a moment. When you think about, I mean, as a lifelong New Yorker, it's been amazing to see the changes that have happened in terms of the entrepreneurship scene in town. For many years now, startups that really are a couple of people on a laptop have had many options. WeWork has kind of transformed that industry. For companies where you need more than just a laptop, where do you go? And do you think the next company you start will be here in New York City?
So I currently go to California, which is the way it is. But the next company will be at Harlem Biospace.
So my first one was at Audubon. Skinetics was at Audubon. The second one left before it got housed. And the third one, Rapunzel's actually at Harlem Biospace. But we have so many options now. There's the NYU Incubator, there's Alexandria, there's so much more. So if your needs are simple, cell culture and a centrifuge, you can actually find that now. And again, Harlem Biospace, a shout out to Sam Sia and the colleagues here for getting that off the ground. So we should take pride, I think, in that too. So there's a lot more available now. And each of those little incubators has their own receptions and networking and events that you can avail yourself of, all for free. So you can crash a lot of parties and meet a lot of people without having to be a tenant. I shouldn't say that.
Well done.
No, but it is, for those of you, Harlem Biospace is a biotech incubator started by Sam Sia from the Department of Biomedical Engineering. And it's, again, right down the hill in West Harlem. There's also now a whole new crop of incubators that have popped up. So J Labs, which is Johnson & Johnson's innovation company, has started a biotech incubator down by Varick Street. Alexandria Real Estate, which is one of the world's largest biotech real estate companies, has one of their incubators down on the Far East Side. So there's an increasing number of options, and I'm sure I'm forgetting a couple, which I'll regret later when they yell at me. So that's great. It's also worth noting the mayor last year announced $500 million towards promoting the early stage commercial life sciences in New York City. So there'll be a whole bunch of new incubators opening through that. And the governor then announced $650 million to do the exact same thing, but across the state. So there's tons of resources coming in this direction. In terms of engineering, though, outside the life sciences,
a lot of application for life science. So.
Yes, that's true. That's true. But for Voyant, do you think this is like a company that could live in New York?
Absolutely, absolutely. I think when you look at all companies that are in this subspace, they're all over the place.
Okay.
So absolutely.
Okay. I want to, 5 minutes left. Okay, so I've got time for 2 more questions. One is, one of the things we hear from the PhD students who work in our office, we have a program called the CTV Fellows Program, where we have 35 PhD graduate students from around the university who work part-time in our office, helping to help the entrepreneurs get these things going. One of the things we hear from PhDs who leave and join a startup is that the pace of life and the way things are done in startups in some ways is very similar to their life in the lab, and in some ways is the exact opposite. And they need to unlearn all the non-technical stuff that they've been doing, all the reliance on proving the data before getting all the data before making a decision, taking 5 years to think about something before getting your PhD. How's your experience been in balancing the mindset of being a scientist with the pace of being involved with the startup?
So I can, in my case, it's actually simple, because if the 5 years it took, or the more than 5 years it took to work out what works, works. And so you don't need another 5 years to prove it works. You can plug and play, and that is that's very good. That feels very good, because I'd hate to be in a situation that I don't have the total control over the environment in that new startup, where other people will do it their way and it doesn't work, and then I'm stuck with that. So in my case, this fear doesn't apply, thank God, you know.
So I'm a little bit lucky there, I think, because in many ways, both at the lab and in the startups, we're constrained by the rate of hair growth. We can't make hair grow any faster than it's going to grow. So the data takes as long as it takes in either context. But the thing that's been surprising to me is the sense of urgency around clinical development milestones. So once your stuff gets into a phase 1 study in the clinic, boy, does that have a life of its own. The timelines are rigid and adhered to like to the day. So, you know, in science, if you miss an NIH deadline, you wait 3 months and there's another one, right? It doesn't work that way in their world. So the rigid nature of some of the business development and clinical development milestones, to me, was a real eye-opener. There's just not a lot of room there for wiggling. And again, the level of burden of proof is different. You know, it's what you go out and pitch to investors. It doesn't have to be the standard of publishing and sell. That's different for us.
I mean, it's driven by a return of investment. And that's different.
Yeah, makes sense. So one last question. I would be remiss sharing the stage with 3 astoundingly accomplished female scientists and entrepreneurs, which is unfortunately still fairly rare in the tech entrepreneurship world. Any advice you have either for women entrepreneurs and scientists that you could pass along? I know we have a group here called the Columbia Women Inventors Network, which is trying to get folks together for conversation, networking, and learnings. But any advice you'd pass along, or just on entrepreneurship in general? Anything you think, 1 or 2 things you think the audience should take away from this?
Well, I would say that the one thing or at least I myself strived to is focus on the numbers, choose a technology that is very clear-cut, meaning these are the parameters that needs to be achieved, and then you actually do it. And I'm fortunate that in engineering, even applied engineering to all kind of different areas, this can be done. But I think also in life science, you can also do that, and that kind of relieves all subjectiveness, or maybe politics, or maybe bias that we all as a society has. It relieves that. Either if you have the numbers, this is it.
Yeah, I second this. That's, I think, one of the big advantages actually in these more scientific worlds. If it works, it works, and if it doesn't, it doesn't, and it doesn't really matter what gender you have. If you have something that is irresistible, go for it, and it'll take you there, you know. And the new generations, much younger than me, they will, they will take that much lighter. I expect, I hope, so. It's also a good opportunity if you I want to point that out. There are not too many role models, so you can just do as you like. That's also good.
I get into trouble when I do as I like.
Go for it.
I guess you don't want our Me Too stories, right? We only have 1 minute left on the panel, so we'll skip the Me Too stories.
We all have many.
We do have the Me Too stories, but not today, maybe. So I would say that, so I think there's a tendency in this, I've seen this in the rooms, for our male scientist colleagues, I think there's a tendency by the business partners to need to school us, feel the need to sort of, treat us like we don't know what we're doing in the business world, which is the truth for most of it, at least for your first deal. But by the second deal, you need to be really good at your game. And so I think the power of learning how to leverage being underestimated is very important. So we're all underestimated for a living. I'm speaking for all 3 of us because I know that that's true for them. But learning how to capture that being underestimated and turn it around and use it like Wonder Woman right back at them is your most powerful weapon. So by the time you do your second deal, you got to have your golden lasso ready. So it's very hard the first time because you really don't know what you're doing. But by the second time, whether your first deal goes good, bad, or in the middle, you will learn some really hard truths and lessons from that. The best thing you can do with that, at least in my experience, having gone through round 2 and now round 3, is to let them think you're underprepared or let them underestimate at their own peril. And by the time you negotiate that second deal, they should be cowering in the corner.
Great. Well, if this is a topic of interest to the audience and you want to learn more, there's some great, oh, sorry, not that specific one. There's plenty on that as well. But if in general the topic of how these inventions leave the lab and become technologies that save and improve people's lives and how you can play a role in that, there's a number of classes at the university. There's Research to Revenue, which is a joint class between the business school and the engineering school that's open to the whole university. There's Launching New Ventures in the business school. I teach a class next spring called Intellectual Property for Entrepreneurs, which talks about negotiations and startup launches and commercial side as well. So there's lots to dig into. Entrepreneurship.columbia.edu, which the entrepreneurship organization runs. And please join me in thanking our amazing panelists today. It's been great.